Thank you so
we
much.
have to rush to the next speaker. We're going to go to a little more different area. This is also affecting something that is really of very crucial importance for humanity today. Humanity and also all the living organisms on the planet, which is the multi-drug resistance that has been developed because of the, I would say, maluse of drugs that we have done in the past. So there is a development of bioinformatics tools that will deal with all of these things, and this would be presented by Emiliano Barreto-Hernandez. He is from the university, the National University of Colombia, and he is located in Bogota. So please, Emiliano, share your screen and start your presentation.
Thank you very much, Eric. Thanks for the introduction. Let me try to start the presentation. The idea is try to introduce this new term that is precision epidemiology that is applied now to the multi-drug resistant bacteria, and how the tools that are developing around the world in special for processing a big amount of data and for processing the new sequences that are produced by the new sequencing systems are involved in this precision epidemiology process. Something very important, I maybe miss Leon Institute, an important institution here, is that it's a process that we started close to 10 years ago. And in that process, the EMBnet has been very important because the connection between groups that are involved in this project. I have to thank to the Fribourg University, the Swiss Institute of Bioinformatics, the HAH from Belgium that helped us this year with a student that worked in this project. And I have to emphasize the contribution of the EMBnet and the connection that is possible through this network. In this presentation, I will try to go not in complete detail of the system, but I want to do an introduction and present the genome information management system that we are developing in the last years, some results and some general conclusions. And the first thing is how you can understand what is the precision epidemiology. Epidemiology is something that is used for elucidate the source, the time, and the transition and the spread of the diseases. But when you apply this to infection disease control, you need a way that you can really identify and type in the bacteria or the microorganisms that are involved in this infection disease. That precision epidemiology was built in base of the new generation sequencing. And the important thing is that it's possible follow with a very specific approach the diseases and try to control them and try to control not only when appear in an individual in specific individuals in the society, but control them at the population level. The thing is, as Eric mentioned before, that the infection disease could be a problem in less than 30 years since now. There is a projection of what could be the people who will die for this kind of disease. And maybe in the 2050, this disease could be the most dangerous one, because it's projected that more than 10 people million die every year for this kind of diseases for that time. Now, well, the World Health Organization is trying since more than 30 years to do something with this resistance to antibiotics that present some bacteria. And they try since more than 30 years to have a system and a surveillance system that harmonize the global information and put some standards for maintain that information. The most important part here, like you for sure have seen with the COVID, is that you have some kind of central repositories that collect the data, not only the information from the patients or the clinical information and microbiology information, the systems that allow to maintain the molecular information that is possible at the moment. And in that way, to have an idea how extend or how different are the different microorganisms that produce this type of problems, or to have a global report with correct data that allow detect the resistance and detect how the resistance is spread around the world, and inform to the different organizations related with this for control this in every country, how to do some kind of prevention or some kind of control, or how to use the treatments available for this kind of diseases. You can see this in this map from the World Health Organization, how the glass system that is was an initiative from 3 years ago is starting to be implemented around the world. There are many countries involved in this, and the idea is that using the interconnection and the standards, we can have a kind of federated repository of the data related with this kind of disease around the world. You see, for example, here, Colombia is not in the process. We are starting to work with our National Institute of Health for food standards that are in this GLASS project. And the tools that I will try to explain you today are related with this connection with the GLASS initiative from the WHO. Anyway, when you try to do this, connect this disease and the precision epidemiology, the method that appeared as a first tool is the new sequencing techniques. Here you can see how appeared in the last 20 years, and you can see the most important companies that produce this, and especially Illumina, that is the standard of the company that have more part of the market in sequencing. But the important here is that it's possible in a very low price to have the complete sequence of the genome of these bacteria, because they are the small genomes. And with that, you can really produce a precision detection, and you can follow these bacteria easy and connect what is the behavior around one institution or in a city or in a country, and if you connect these things using initiative like GLASS, you can see what is the process around the world. Why is important? Because in this moment, the whole genome sequencing using this new generation techniques for sequencing really is economic, is fast, is the speed every day increase for obtaining this kind of data. The errors are less and less every day. And in general, the important thing is because you have the complete information of the, at least the things that are coded in the DNA. For the bioinformatics part, you can see that there is an effort around the world, everywhere, that is producing new tools, improve the tools for analyze and process this kind of data and connect this kind of data with the other kind of data that is important in this process. With that background, we try to produce our genome information management system. We started as every other lab in the world, trying to sequence in a few isolates from the hospitals or from the society, and started to create some tools that allow us to manage this genomic information. But the thing is that you need more than only the genomic information in the system. You need to use the complete set of data for the exactly identification of the bacteria, the typing of that bacteria, and connect that genomic or molecular information with the antibiotic resistance or virulence of that bacteria, and try to follow in a specific, with a precision, how that bacteria is appear or how that bacteria moves in different moments in the society. For that, in that system, we integrate demographic data. We can see some of the principal data, but not the system is not limited to this kind of data. And we put the genomic and the microbial and the clinical data. Well, I have to point out here that the clinical data is some kind of challenge, because the limitations for the patient information. We have special projects with inform. I don't know in English that word, but it's in Espanol is consentimiento informado, is the approval from the patient to use the samples. And with that, we integrate the system using web app. We try to integrate a web application because we wanted that the application could be accessed by the people in the hospital and other people around us. And that application was made based off Java, some kind of Java called Grails. That Grails has a specific way to build the application that is three different layers. The one layer is the view layer that is connected with the users. The other is the controller layer that is pass the data from the user to the model layer that is responsible for access to the database. We use for this BioPython and BioPerl for develop scripts that are included and connected with this web application, and we use MySQL database for maintain the data, to organize data. The idea was in try, since we collect the sample, have a way to put the demographic information and the clinical information and the microbial information and the genomics in a computer when we create an interface for manage these kind of things. And inside, we create a pipeline that can process with different tools. Normally, the tools that are most more used in this moment in pipeline for the NGS data, like FASTQ, cycle, spades, or Broca for annotation. And when we use this pipeline, we pass that data to the relational database, and after that we can produce this kind of analysis. We put many of the tools that are available for this kind of analysis, comparative genomics, or extraction of features, or typing, or identification of some features related with resistance or virulence. We are exploring virulence. And with the final result is interface that can be accessed through internet, and we have a module that allows the access. The important thing here is that we have restrictions to a complete set of tools that allow safety management of the data, and especially of the clinical data, because it's restricted because the low restrictions. And with that, you can access, when you pass that access model, you can access the principal modules in our system. There is a module for search the data, or a module that allows to put the data inside the system, and a module that is using for reporting, to analyze or to show certain kind of relation between the data. For example, here we have inside the system where you are putting the data, the possibility to select the tools that you can use for processing your NGS data, and automatically the system use the pipeline and put the information after the processing inside the system.
1 minute.
So it's only 1 minute, sorry. I will go fast. We have a system that these things are with tools that allow to have access to the reports and the analysis, and use databases that we created specifically for certain kind of elements related to resistance or virulence. And we just developed some software for the identification. We have some process that allow to use random forest models, for example, or use the database and comparison for obtain the genes that, or the elements that are related with resistance, other approach for typing and identification of the bacteria related with resistance, workflows that allow to separate the chromosome and the plasmid part of the sequence when you obtain the final sequence from the Illumina, for example, and you need to check which is plasmid or which is chromosome. And with that, we can produce the reports that allow to check in a specific way the epidemiology of these isolates. Here an example with many isolates from Colombia. Or here are the information related with any possible genomic alimentation related with antibiotic resistance and the difference between strains. And when you need to check in more detail for a specific strain, you can arrive to these comparisons that allow to see the difference between states that are clonal related, but they produce a different behavior in the hospitals. And then finally, we created an integrated system that joined the clinical, microbial, and molecular data and allows follow in a precision way the bacterial resistance. And this thing, I have to mention that this SIG is a prototype. We are trying to improve every day with the collaboration with many people. And the most important part is it's easy to integrate wherever analytical or bioinformatics tools available for analysis or for the integration of this data. Very important is the sponsors for this project could be our national agency called Ciencias, the National University, the PFL in Lausanne, and I have to thank the people that is involved. Here is only a few people that is involved in the project at this moment. Thanks very much.
Thank you very much, Emiliano. Very interesting. I think this is something that people will have a lot of questions.